Feline Immunodeficiency virus, or FIV, is an enveloped RNA virus. It belongs to the lentivirus genus of the Retroviridae family, and as its name suggests, it causes disease in feline species, both domestic and wild. It is found all over the globe, with 7 different subtypes affecting more than 14.5 million pet cats worldwide, and even higher numbers of feral and wild cats. Cats are most likely to contract FIV when bitten by an infected cat during a fight. Since FIV is shed in high concentration in the saliva, this is considered the primary mode of transmission, although other forms of transmission, such as from mother to kitten, is also possible. FIV is not environmentally stable, as its lipid envelope is easily breached by detergents, heat, and desiccation. Free-roaming male cats are particularly at risk of contracting FIV, as their territorial behaviour, especially during mating season, increases their chances of being bitten. An FIV infection is typically categorized into one of 3 phases. Stage 1 is a primary infection. As virus replicates, infected cells are lysed, leading to a marked viremia and signs of malaise. CD4-positive T lymphocytes are most notably affected by the invading virus. However, as FIV uses chemokine receptors to invade cells, it can infect a number of additional cell types which do not display CD4, such as macrophages, monocytes, and dendritic cells. A lymphadenopathy may persist for multiple weeks, as well as a neutropenia. Stage 2 is an asymptomatic phase, where the cat, though still infected, will appear clinically healthy. The host's immune system reduces viral load, but the high rate of mutations results in varying surface proteins to evade the immune system. This phenomenon is known as antigenic drift, and is what allows FIV to persist as a chronic infection as it escapes the recognition of host antibodies. This stage can last for years, or in some cases indefinitely. Stage 3 is the terminally infective stage. Due to the immunodeficiency caused by the virus, an infected cat is more susceptible to secondary infections, which result in clinical disease. Clinical signs are therefore variable. A cat in this stage of disease may present with a range of signs, such as cachexia, depression, gingivitis, stomatitis, neoplasia, and neuropathological disorders. Currently, there are no effective treatments of FIV, so preventative measures are of great importance. Commercial vaccines such as Fellovax FIV are available in Australia. However, the efficacy of the vaccine is doubtful, meaning that a vaccine should not solely be relied upon to prevent FIV infection. Where possible, cats should be kept indoors to prevent the spread of infection. Desexing may also be an effective strategy to prevent wandering or aggressive behaviours. Antibody tests such as Witness and Antigen Rapid are useful to diagnose FIV, as they can differentiate between antibodies generated from a vaccine and those generated from an infection. Such tests should always be considered with clinical signs and patient history in mind.